Okay, so good afternoon. Welcome to the week two discussion session for the immunology course. People who were able to attend last week's lecture, you're already familiar with the idea that we are trying to follow during these discussion sessions. Just excuse me. It's sharing. Just give me a moment. The idea during these discussion sessions are to go over the topics that have already been discussed as part of the lectures that you had followed for each week. Along with that, I will be adding some extra information regarding those same topics, some additional information, some extra schematics, for example, and we'll try to conclude with some sample questions based on the topics that you have covered for that week, based on the questions that are part of your assignment. However, we will not be discussing the assignments right now. We will discuss them after the deadline is over. We'll directly take up the questions. And since the deadline is on 7th, we'll take both week one and week two questions together. Apart from that, at any point of time during these lectures or these discussion sessions, if you have any query that you want to be addressed, please bring that up. We'll try to either sort it out in this session itself, or if it is a topic that I don't have slides for or something that I myself have to read up on, we'll take it up in the next discussion session. So that is the idea. Now, while in the last week, most of the information that I had included in my slides had been from just one textbook, which was Kobe Immunology. As you already know, Professor Shudeep had suggested multiple books. You can refer to any of them. But today, specifically, I have included multiple schematics from research papers, review research papers, where they summarize articles based on what is currently being looked into on each field. As you already know, today's topic is majorly centered around innate immunity. Now, this week, all of the lectures were very information intensive, so I will not be going into each and every topic in that much detail. Professor Agnew has already looked into in extensive details, but yes, we will be brushing up on some of the topics that he has covered. Um, I have given reference for each of the articles that I've cited the schematics from. So if you want to read up, go into more depth about it or read up on it, you can certainly go to the links. I can even provide the link separately, or you can directly refer to the article for your own understanding. With this in mind, we will start today's discussion section. Let me again reiterate that if there is any doubt at any point of time, please bring it up. I will either note it or will try to address it immediately if it is something that I'm also clear about. So with this in mind, we start off the discussion for today. In the last class, if you remember, we had briefly looked into the differences between innate and adaptive immunity. We had talked about how it is the first line of defense, how there are multiple sub-steps towards innate immunity, such as the physical, physiological, or anatomical barriers, and the different receptors that operate. So we had a summarized table. I can go to it if that is screen is freezing. Just give me a moment. Yeah. We had looked at the different types of barriers that exist, which were non-specific host defenses, such as the anatomic barrier, skin, mucous membrane, etc. The physiological barriers like temperature, low pH, chemical mediators, etc. And phagocytic, endocytic barriers, and finally the inflammatory barrier. So this was just an introduction into the topics that are getting covered in this week's lectures. So with this in mind, we will start week two. So first of all, let us okay, my slides today also might look very information intensive, but I've kept it that way so that it's not just a schematic that is confusing to somebody who is reading up on it, but instead you can actually understand what is being said. What you see here is a summary of how innate immunity works. These are the different mechanisms, molecular mechanisms, or we can say defense mechanisms that operate as part of the innate immune defense. So, what you see here are the subclasses. First is PAMP-triggered immunity or ETI, effector-triggered immunity or ETI, recognition of absence of self, which we have previously termed as self-non-self recognition, then recognition of altered self. So what is the difference between these two? When we talk about absence of self, it means something which has come from outside of an individual's or from the host body. So, for example, a microbial infection is the absence of self or it is non-self. In contrast, when we talk about altered self, it means, for example, an autoimmune cell, a cancerous cell, where the cell belongs to the host body but has undergone some sort of changes which has led to an alteration that needs to be removed by the immune system. Then comes damage recognition and finally surveillance. We'll look at each of them one by one. So first, what you see on the right, refer to the legend and the figure simultaneously. I'm not able to switch on the laser point. Okay, yeah, it's working now. So first is the PAMPs triggered immunity. So PAMPs are pathogen-associated membrane proteins. These are, as the name suggests, they are proteins present or antigens present on the cell surface, and these are very crucial towards the recognition of a foreign body. So what you see here, PAMs, otherwise known as MAMS, M stands for microbes. So these are recognized by host pattern recognition receptors or PRRs, which are able to trigger an immune response. So what these pathogen recognition receptors do is that they bind directly to the pathogen, as you can see here. Pink represents a microbe or a pathogen. These Y, U, or V-shaped structures are the receptors. And these are microbial molecules. When we say microbial molecules, they're microbe cell surface entities, antigens, etc. And what you see in blue are host molecules. So this recognition by MAMS or PAMS is able to trigger a response, and that response is initiated by the host surface proteins known as PRRs. Second is the effector triggered immunity. So as the name suggests, this is not triggered directly by the pathogen, but is instead triggered by something which is released by the pathogen. So for example, here you see the microbe releasing certain effectors. So these effectors can often be called as virulence factors, which is an antigen which can be recognized directly by the immune system as foreign. So host receptors are capable of sensing these virulence factors, or more frequently, they are also called as guard proteins that can sense that there is a virulence factor which is modifying something within the host. So this allows these host guard proteins to identify, recognize, and trigger an immune response against the same. Host guard proteins can either be extracellular, transmembrane, or intracellular, depending upon how it is functioning. The third factor, as I have already discussed, is the recognition of absence of self. So the immune system becomes active when there is a constitutive signal provided by host cells and is not detected by host receptors, which means that it is a foreign origin or something which has come from outside of the body. So these blue molecules that you see are host, and therefore they are able to bind to the host receptor, but dark blue molecules, but something which is not belonging to the host or which is not intrinsic to the host will be recognized as non-self. Then comes recognition of altered self. I've already discussed it in some detail. I gave you the example of autoimmune cells, but instead, sometimes it can so happen that these pathogens are intracellular in nature. So they have invaded into a host cell, and then they are altering it somehow, which is causing which is triggering the immune response. Then comes damage recognition. So damage recognition is carried out by damage associated molecular pattern receptors or DAMPs, just another subclass of receptors. They are able to sense some molecules which are released when a cell is damaged. So what you see here is an intact cell. It has undergone some sort of damage. For whatever reason, it is damaged either by a microbial pathogen. It has undergone some changes in its genome, which is causing it to die or undergo apoptosis. If you're not familiar with it, apoptosis is the process of programmed cell death, where a cell with complete knowledge of the fact that it is undergoing a process which is compromising its survival, it chooses or it opts for death. There is a very well-regulated process which triggers death in such cells, and it is a programmed process, hence leading to the name PCD or programmed cell death, otherwise known as apoptosis. So when such cells undergo such processes where they are dying, then in such cases, there can be release of certain factors which indicate that there is damage occurring to the host system because of some invasion that has occurred, and that damage can be recognized by these receptors known as DAMPs. And finally, what we see as part of the innate immunity is the surveillance mechanism. So innate immune responses can be triggered by stress pathways that interpret the rupture of cellular homeostasis as a recognition of infection. These sensing mechanisms are either direct or indirect, depending upon how the activities can be sensed. So what we see here are that under default circumstances, the immune system regulates a surveillance mechanism. Surveillance basically means that you are keeping a check at all times within the system to identify whether there is something foreign which is roaming around in the body or not. In case something is recognized, it will trigger a cascade that will identify that there is a foreign invasion and this needs to be removed immediately. So these are broadly the different mechanisms or just the subtypes of different mechanisms that are operating all together to keep innate immunity intact within any living organism. So that's how it works. So keeping this in mind, what we now look at are the different comparisons between innate and adaptive immune system. So what you see here are the different properties. It has been classified based on properties. So innate immune system has receptors that are already pre-programmed. There is no change which is occurring there. No rearrangement is required. However, when we talk about adaptive immune system, which we'll get into much detail, the gene segment rearrangement is a very, very crucial part of it. And rearrangement is necessary in order to allow that diversity that exists within the immune system for recognition of pathogens of all different types. Which is something you'll come about when you understand how antibodies are made, how gene segment rearrangement occurs. I think I mentioned it in the previous discussion session also that there was a Nobel Prize which was awarded for discovery of that process because it is that crucial and that important and that complicated a process that its study was extremely relevant as a contribution towards science. So these were about the receptors. Coming to the distribution, innate immune system is non-clonal, which means that all cells of a particular class are identical. Adaptive immune system has clonal cells where each subclass has distinct receptors and they can be clustered based on the type of receptors that they have. Third comes recognition. When we say recognition, the innate immune system is able to recognize some conserved molecular patterns, like we just talked about the different pattern recognition receptors which operate in that case. Here you see some examples like LPS is for the lipopolysaccharide layer, LTA is lipoteichoic acid. These are basically small antigens or small surface molecules which are present on different types of microbes. So the innate immune system is able to recognize these different types of unfamiliar patterns and is able to trigger a response against it. Then come mannans and glycans also. These are sugar molecules which are present on the surface of microbes or pathogens in general. On the other hand, for the adaptive immune system, molecular structure is more relevant. Proteins, peptides, carbohydrates, etc. Those are looked into and those are more relevant. Self non-self discrimination is perfect for the immune system. It is selected. It has evolved over time to become very, very accurate. It's able to clearly distinguish between self and non-self. However, coming to the adaptive immune system, it is not always perfect. And when compromised, it can lead to autoimmune disorders. Talking about the action time, it is immediate, which is why we call the innate immune system as the first line of defense, whereas adaptive has delayed activation of these effectors. The response is mediated for the innate immune system by co-stimulatory molecules, cytokines, and chemokines. These definitions have already been elaborated in the lectures themselves, that cytokines are molecules that mediate cell-cell communication. Chemokines are a subclass of cytokines. They are more specifically serving as chemoattractants. When we say attractants, it means that their release is causing the movement of cells towards those molecules, and that is why they're able to drive or they're able to control cell migration. Yeah. So That is the difference. That's the difference between cytokines and chemokines. Some examples have been given here. IL-1 beta, IL-6. IL stands for interleukins. These are different types of cytokines. There are probably some dedicated lectures for cytokines as well. The entire immune system is extremely heavily dependent upon the way these cytokines and interferons they work. These are the molecules which convey signal from one particular component of the immune system to another. You can consider it equivalent to the role of hormones in the endocrine system. So the way in which different organs are able to communicate with each other, starting from the hypothalamus to the pituitary, from there to say the adrenal glands or any other gland. So that entire axis is entirely controlled by hormones. Similar to that, exactly drawing the same analogy for the immune system, cytokines play that crucial a role. So for innate immune system particularly, it's extremely crucial. Coming to the adaptive immune system, there is clonal expansion or clonal energy. Clonal energy is also it's a slightly complicated concept we can come to a little later. IL-2 is majorly the interleukin that works for adaptive immune system, and there are some effector cytokines like interferon gamma and IL-4 which are operational there. So this was just to draw a comparison between the entities that play a role when it comes to innate immunity. Now, what you see here at the bottom shows the different types of cells which have a role to play in the innate and adaptive immune system. So, as we have already discussed, the cell that is believed to overlap between these two subtypes are the dendritic cells, but majorly the innate immune cell is dependent upon, first and foremost, what you see highlighted the most is the neutrophil. Basophils, eosinophils, monocyte macrophages, mast cells, and natural killer cells are all part of the innate immune system. They are all WBCs, as we commonly know, white blood cells. And the adaptive immune system is extremely heavily dependent upon the T lymphocytes and B lymphocytes. But their role is not entirely exclusive. There is a crosstalk that occurs between the innate and adaptive immune system as well, which is something you've already learned about in the lectures. So with this in mind, we move forward and we try to understand what are the modulators. So again, because this is a summary, I've not gotten into excessive details about them, but just a comparative study of these so that it will be easier for you to even make notes or just conceptually remember the different subtypes and the different receptors which are operational here. So the types of, we talked about pattern recognition receptors being very relevant towards the functioning of the innate immune system. But when we talk about the pattern recognition receptors, they are of different subtypes. The first type you see are TLRs, toll-like receptors. Again, keep in mind, it's not a single receptor. They are a family of receptors, each of which each TLR is responsible for triggering a different type of pathway. So depending upon the type of pathogen that has come in, the type of surface antigen that it carries, a different TLR would be able to bind to it and would be able to trigger an immune response in a different manner. So toll-like receptors are localized in the plasma membrane. Their ligands are lipoproteins, DNA, RNA, endotoxin, or endogenous danger signals. The source of the ligand would be either from bacteria, viruses, parasites, or can be a cell also. NLR are not like receptors. Their localization is in the cytoplasm. So you see how it's different, right? For cells that are, or for pathogens that are not intracellular in nature, that are only present in circulation, those would be recognized by plasma membrane toll-like receptors, but those that are able to invade into the cell are able to come into the cytoplasm. In that case, NLR and in fact another class of receptors as well, known as RLRs, are able to take over. So not like receptors are able to sense, again, they are also able to sense endogenous danger signals and also muramyl dipeptides. This would be an interesting thing for you to look up. You can see where muramyl dipeptides are generated, which or let's put it this way, what would be the source of muramyl dipeptides? I will mention that it comes from bacteria, but maybe you can look up where it is present in the bacteria and why can it not necessarily be recognized by surface receptors? Why is it coming inside? Third are CLRs or C-type lectin receptors, again present on the plasma membrane. They are able to bind to beta glucans, which are present on fungi. And RLRs or retinoic acid inducible gene 1-like receptors are cytoplasmic in nature. They are able to bind to double-stranded RNA, which comes from RNA viruses. So you see the innate immune system also has a basic surveillance mechanism where it can tackle ligands from different sources, including bacteria, fungi, and viruses as well. So, and parasites as well. So, different receptors have been kept in position in order to tackle different types of problems. Now, coming to the ABCs of innate immunity, these are just a summary of the abbreviation so that it's easier for you to remember. We talked about pattern recognition receptors, pathogen-associated molecular patterns, danger-associated or in fact damage-associated molecular patterns are for DAMs. MyD88 is a product of myeloid differentiation gene 88. You are not necessarily coming to it. It's just a table that was already available. So some terms might be unfamiliar. NF-kappa B is something you'll come to later. It is nuclear factor kappa B. It's a transcriptional factor and it has huge roles to play in cancer also, not just immunity. TLR are toll-like receptors, like we already mentioned. NLR, CLR, and RLR are something we've already discussed. So these are just some basic terminologies which are modulating innate immunity. And it's just giving you a comprehensive idea of what exactly you need to keep in mind. Now coming to more details about these. So first of all, what you see, it is a schematic representation of the way in which innate immunity is being modulated. I'm in today's discussion session, the slides I have with me will be focusing majorly on innate immunity and less focuses on T cell and B cell maturation, because those processes are very you can pick up a book and you can read it from there. I will summarize it as well. But yeah, I would suggest you can refer to the lectures because they have been extensively discussed there, and also any book would be able to clear it up for you. But yeah, because innate immunity is a vast topic, I am focusing more on the way in which it is regulated. So yeah, keeping that in mind, what we see here is a schematic representation of the way innate immunity is being modulated. So TLRs, like I mentioned, toll-like receptors, were first identified based on the toll protein in Drosophila. They are the first PRRs. There's a typo here. Pattern recognition receptors, or PRRs, that were identified, and they have been studied in across the animal kingdom, or across across organisms, across species. The members of these family are all glycoproteins. You can see the different types of localization that they have. So they are immediately present on the plasma membrane. You don't see them detached from it. Either they are transmembrane in nature for TLR 1, 2, and 4, or TLR 7, 8, 9, which are present on the cytoplasmic leaflet of the plasma membrane, but they're still attached to the plasma membrane. Then you have nod-like receptors here, and you have C-type lectin receptors, which are also transmembrane in nature. So these TLRs, or toll-like receptors, they are all glycoproteins, and the N-terminus has an leucine-rich repeat motif with substrate binding ability towards the outer side. The other side is homogeneous to the IL-1 receptor. This is not relevant. It is majorly the reason for them to be transmembrane is that they would be able to bind pathogens or pathogen surface molecules on the extracellular side, and through their intracellular domains, they would be able to convey that signal downstream. So that is the way in which it works. The second subclass that you see here are for the nod-like receptors, which we just discussed. They belong to the cytoplasmic PRRs, and they also contain many different subtypes. And they structurally have three domains. They have a leucine-rich repeat domain, which you see in this sort of a zigzag structure that you can see. They have a nod and a caspase recruitment domain, or CART. LRR domain is at the C-terminus of the NLRs, and they can recognize the ligand. Nod is the middle domain, belong to the superfamily. So this is just extra information. I'm not reading it out. You can read it up for your own understanding. And the third type are the C-type lectin receptors, which, as we saw here, they are majorly responsible for identifying beta-glucans present on the surface of fungi. So what this image basically summarizes is that you have host innate immune cells, which includes macrophages, dendritic cells, neutrophils, and natural killer cells. You have host pattern recognition receptors, which are of these three major subclasses, like I just discussed, in response to identification of the pathogen that has come in and the innate immune cells that have acted. There are also some defense mechanisms that are being facilitated by certain processes. What are those processes? Either there can be phagocytosis, which means that it would be engulfed. There can be autophagy, which is a way of cell death as well, but again that is different from apoptosis. You can look it up, or we'll take it up in the next class. The differences between necrosis, apoptosis, and autophagy. They are all methods or processes of cell death. The way in which they are regulated and the way in which they are coordinated, or under which situation a cell will choose to undergo what. These are all very, very stark differences between these processes, and they are very interesting areas of research even today. So this we can take up maybe in the next class. In the meantime, you can look into it for your own reference. And what you see here is a representation of an inflammasome, which is a moiety or an entity which is able to trigger inflammation. So this is the way in which the host immune cells, the recognition receptors, and the defense functions all coordinate with each other. Again, let me reiterate, if something is not clear in the slides, please let me know. They are very writing intensive today, but that is for your own good, and so that it's easier to remember the information that I'm saying while I'm explaining. Now coming to the cells of the innate immune system. So we have already seen that majorly there are some cell types which are operational. Excuse me. Some cell types which are operational, and that includes the monocytes, macrophages, neutrophils, which are the first line of defense, the dendritic cells, natural killer cells. These are operational, but the phagocytic cells, particularly if we have to divide them into subclasses, there are some cells which are phagocytic in nature. The name indicates that these are cells which are able to engulf or eat up a pathogen that has come in. And there are some non-phagocytic cells as well. How do they differ? We'll come to that. When we talk about the phagocytic cells of the immune system, they are of three types: macrophages, neutrophils, and monocytes. Macrophages and monocytes obviously have a relation between them. If you remember, we had talked about it, that monocyte macrophage transition occurs. So blood monocytes, when cells are circulating within blood, or when they're in circulation, they are monocytes. But when they undergo, when immune defense mechanisms have been triggered, then they differentiate and they become macrophages, and they are able to enter into tissues. So both these cells are phagocytic in nature. The cell type is agranulocyte, which means that if you observe them under the microscope, they do not have granules present within them. That is how they are classified as granulocyte and agranulocyte. Location for monocytes is in blood. Like I said, they're circulating within blood. Neutrophils are also present within blood, but they are granulocytic in nature. Macrophages, on the other hand, are tissue macrophages, and therefore they're either present in some organ, or they're present within some body cavity, etc. So these cells are phagocytic in nature, and that is their contribution towards the innate immune response. So that is how this entire process is working. Apart from these phagocytic cells, when we talk about phagocytic cells, it obviously makes sense that, yeah, there is a pathogen that has come in, pattern recognition receptors are able to identify it, and then these cells are able to swallow it up and are able to degrade it and able to remove it or clear it from the system. But what do non-phagocytic cells do? We said that both of them are operational when it comes to the innate immune system. The non-phagocytic cells of the innate immune system are NK cells. As you already know, they are natural killer cells. So what do they do? You look at here, the schematic here gives you an overview of the way in which NK cells work, and you see that they are able to regulate a lot of different types of processes, and in fact, a lot of different types of cells as well. Let's come to that. First, let us focus on the legend. So NK cells are a type of lymphocytes that have the ability to induce apoptosis, that is programmed cell death, in cells infected with intracellular pathogens such as obligate intracellular bacteria and viruses. These cells can induce apoptosis, in which a cascade of events inside the cell causes its own death by either of two mechanisms. So what did we understand here? NK cells are majorly responsible for tackling intracellular pathogens. In particular, it mentions two examples: intracellular bacteria or viruses, which are able to enter into the cell. In case it is not clear to anybody, intracellular pathogens are only pathogenic in nature when whatever component of theirs is able to cause disease enters into the cell. Their actual pathogenicity comes upon after that situation. Somebody is unmuted. If you have a question, please raise your hand or just mention that you want to ask something. So yeah. So what they eventually do is that they trigger apoptosis or they trigger cell death, and the mechanisms are as follows. First, either they are able to respond to chemical signals and express the Fas ligand. So there is a Fas signaling pathway, which is a death signaling pathway that can operate inside the cell. So Fas is a ligand that binds to Fas receptor, which has some death domains, which triggers to the cell that the cell is not healthy and it needs to die. So that is the way in which NK cells are able to do this, and they are able to increase the expression of the Fas ligand. It's a surface molecule, like I mentioned, that binds to the Fas ligand on the surface of the infected cells, sending it apoptotic signals, killing the cell and the pathogen within it. Or the alternative mechanism is that the granules of NK cells come into play. Like you just remember, I said that granulocytes and agranulocytes are both subtypes of the innate immune cells. When we talk about granulocytes, why are they present? That's because they contain certain digestive substances, which are commonly known as porphyrins and granzymes. What is porphyrin? Porphyrin is a protein which is able to form pores in the membranes of infected cells. Now, for any cell, its integrity, its structural integrity is very critical. When we say structural integrity, it means that the only barrier that exists between the cells inside and the cells outside is its membrane, right? If it is a plant, then it would have a cell wall, or if it's a bacterial cell, it would have a bacterial cell wall. Or in case of some fungi, some certain parasites, they have a slime layer which is present around them. So those are the self and non-self physical distinctions that are present. Now, when you form pores, what happens is that the intracellular contents can be released out into the system, and such a cell will obviously die. So that is the first way in which this works. The most common example that I can give you is that, for example, you would often see that bleach is used whenever we see a lot of growth occurring in certain places. So what bleach does is that it basically forms pores and it kills cells. It also has other operational mechanisms, but that's the easiest way to kill off cells. Or even ethanol, if you've worked in a molecular biology or any for that purpose, any biology lab for that matter, you would find that 70% ethanol is most commonly used for any microbiology system, etc. Also, what it does is ethanol also forms pores, and it causes the cell to eventually die because the cell contents are now released into the exterior. So that's what porphyrin does. But obviously, porphyrin is doing this in a much more regulated manner. It is doing so because the immune system has asked it to go ahead and continue with this process. On the other hand, granzyme is a protein digesting enzyme. The zyme word indicates that it's an enzyme that can digest proteins that enter the cell via the porphyrin pores, and it eventually triggers apoptosis in an intracellular manner. So these are the ways in which NK cells are able to trigger cell death. What you see on the right is a representation of the way in which NK cells are able to regulate processes within the system. So let's just briefly go over it. First of all, what is it doing? It is able to trigger cytotoxicity in multiple cell types. Cytotoxicity of activated macrophages, cytotoxicity via activated T cells. In fact, from T cells, from a premature or a naive T cell to an activated T cell, the conversion involves the use of interferon gamma, which is a type of a cytokine, and so this interferon gamma is getting released by NK cells. In fact, interferon gamma and TNF or tumor necrosis factor is also involved in the conversion of macrophage to activated macrophage, which is able to then cause cell death. It is also able to do this for dendritic cells, where a premature or pro-dendritic cell is converted into an activated dendritic cell, and that also involves these two factors. In fact, these cells, on the other hand, the activated macrophages and activated dendritic cells, release certain other types of cytokines or interleukins in particular, which help in NK cell growth. So what is happening here? When an immune response has been triggered, NK cells have identified that there is some intracellular pathogen and we need to get rid of it. NK cells will start supporting the growth or supporting the activation of cells which will remove that pathogen. What are those cells? T cells, dendritic cells, macrophages. So it is triggering their activation. When activated, these activated cells are releasing certain molecules which are allowing more and more NK cell growth. What is it effectively doing? It is creating a positive feedback loop that more and more NK cells will keep on getting activated. More and more dendritic cells, T cells, and macrophages will further get activated, and the cycle will keep on going until that infection is cleared from the system. After a point, when we see that dendritic cells are no longer able to clear or are no longer needed to clear an infection, then this population will go down and the cell will go to a normal homeostasis condition where it is no longer in a where it does not have a hyperactive immune response ongoing. So this is how regulation is carried out. Yeah. Next, we come to another very, very major function of innate immunity, which has also been discussed, which is inflammation. So inflammation is very commonly interchangeably used with another term, which is known as swelling. But just to remind you that while they physically might look very similar, any person with any swelling might be considered to be, undergoing a very hyperactive immune response, but that's always not the case. I particularly wanted to highlight this because while they're interchangeably used, they're very different. They're quite different processes. So first of all, inflammation. Inflammation is a localized physical process where part of the body becomes red, warm, swollen, or or becomes very painful in response to certain injury or infection that has occurred. On the other hand, swelling is a process that involves abnormal enlargement or abnormal increase in size of a body part. Normally, that occurs due to result of some accumulation of fluid, mostly because your blood cells have rushed there to repair some process. Secondly, what is the cause? Cause of inflammation is because there is either an injury, a wound that has come in, or because there has been some infection by foreign invaders like bacteria or viruses. When we talk about swelling, it can be either due to some injury, some chronic diseases, pregnancy, etc. So it can happen due to a number of reasons. Lifestyle habits, etc. also can be a major factor in it. Diagnosis for inflammation, of course, you would need physical examination, you would need x-rays, blood tests. Major contributor towards identifying a very hyperactive immune response, in particular inflammation, is the increase in this particular protein, CRP. We probably have this next week. I have to check it once, the way in which CRP works. If it is not there, I will definitely include it as part of my lecture, as part of the discussion session, because it is extremely important to understand why CRP is so crucial. In fact, if any of you has ever come across a relative who has been admitted into a hospital for some reason they're getting very high fever and you're not able to diagnose what is the reason behind it, one of the preliminary tests that are carried out after a complete blood count is the CRP test, which is for C-reactive protein, because that's a marker for inflammation. And also ESR, or erythrocyte sedimentation rate, is checked to see whether or not a person is undergoing has a hyperactive immune system as a result of inflammation. Visual examination and other tests like X-ray, CT, etc. can be done for swelling also, but yeah, it's not that crucial a diagnosis. Treatment for inflammation involves very serious medications, which are called as NSAIDs, or non-steroidal anti-inflammatory drugs. Corticosteroids, hydroxychloroquine, biological drugs like different monoclonal antibodies. In case you're not familiar, these drugs that you see that have the name MAB at the end, for example, what you see here is certolizumab or adalimumab, etc. These are all monoclonal antibodies. They are immune therapy strategies. So MAB stands for monoclonal antibody. So these are a subclass of drugs which are increasingly being developed for use in patients. But of course, as you understand, any immunotherapeutic approach is very expensive. So it's not very commercially viable, but yes, research is going on in this direction. So that is the way in which it works. On the other hand, swelling can simply go down if you add ice to the affected area, and you might not even need to take these non-steroidal drugs. But yeah, that's the way in which swelling works. So overall, while they are very similar process, inflammation is regulated, and inflammation has swelling as a symptom, but swelling by itself is not a disease. So that's like the thin line between the two. So then what are the symptoms of localized inflammatory response? Before I get there, you should keep in mind the fact that here we are repeatedly calling it localized. Why so? Because there are certain diseases or certain conditions where patients suffer from a very hyperactive immune response for very long durations. When I say durations, these can be as long as several months, several years also, and those are chronic inflammatory diseases where such a person constantly has a hyperactive immune response when it is not required. Now, as you understand, if a person is constantly having a very increased immune response within their body, it is draining, it is not good, and then it leads to a lot of non-specific responses as well, and it's a condition that affects those people very easily. So that condition would be chronic inflammation. However, when we talk about localized inflammation or localized inflammatory response, response means it is in as an answer to something that has come in. It is an answer to a wound or an injury that a particular individual might have encountered. So the symptoms of this localized inflammation is as follows. The first is swelling, otherwise known as edema, and mostly it is pus-filled swelling. Pus-filled because it is very rich in WBCs at that point. Second comes redness or erythema, heat generation, there's pain, and often there can be some sort of numbness that may or may not occur, and eventually there's loss of function. So while this can be a little confusing that there is pain and there's numbness, both are being considered as symptoms. It depends from case to case, and in some cases it is like, for example, if there is somebody has some inflammation in the wrist, right? So there's a lot of swelling, it's red, etc. There's heat generation, etc. Then such a person might not be able to function very well with their wrist, right? Because it's kind of numb. But if you touch it, there would be pain. So there is pain because the nerve endings there are kind of exposed. So it is painful to touch such a place, but yes, it is also associated with some sort of numbness. So these are the common symptoms or the uh, characteristic symptoms of a localized inflammatory response. The question now comes that what happens during inflammation? So this has already been discussed in the lecture by Professor Ganguly, but I'm just reiterating that because this was an available schematic. Again, this is from Kubi. You can refer to it for your own self. So what you understood from inflammation till now is that it has certain symptoms. Where are those symptoms coming from? It occurs by a very, very, very highly regulated and a very well-programmed process that occurs. What you see here is leukocyte, or in particular neutrophil extravasation, which is a major contributor towards inflammation, is a highly regulated multi-step process. So this you see is a skin barrier. So in case there is tissue damage by some way, say for example, if this is some sort of a nail, or you get a cut from some leaf or thorn or something like that, and the associated bacteria which are present on its surface have breached through all of the dermal protective layers or through multiple layers of the skin, and they have accessed the tissue which is underneath, then you get inflammation. So what you see here are the bacteria which are associated or which were in contact with that source of injury have gotten released into the tissue. What does this do? So what you see first is that this tissue damage is causing a release of vasoactive and chemotactic factors that trigger a local increase in blood flow and capillary permeability. We'll come to it one by one. What is it exactly saying? So tissue damage has occurred or an injury has occurred, and in response to that, vasoactive means those molecules. Vaso means it is part of the vasculature. Vasculature are basically the entire blood vessel system that exists within any individual constitutes their vasculature. So blood vessels are, of course, arteries and veins both. So the vasoactive, which means which will somehow trigger, somehow sensitize the vasculature of an individual. Such factors are known as vasoactive factors. And chemotactic, chemotactic is derived from the process of chemotaxis, which means movement in direction of a chemoattractant. We already talked about a chemoattractant, I think in reference to chemokines a while ago. So that is how vasoactive and chemotactic factors are being released because of some sort of damage that has occurred. And that is triggering a local increase in blood flow and capillary permeability. So why is that necessary? Blood flow needs to be increased because the cells that we talked about, neutrophils, which are the first cells that come into whenever there is a tissue damage and there is an inflammatory response that needs to be generated, the first cells that come into play are the neutrophils. So to increase the propensity or increase the probability that a neutrophil will come at this particular site, or say for example, if you think about any human, one particular person, somebody you know, your friend has extensive surface area over the skin. So wherever that cut has occurred, you need to have a high concentration of neutrophils present there. So to facilitate that, blood vessels or blood flow rather within those blood vessels is increased, and it is also associated with increased capillary permeability. When we say permeability, it means that the barrier nature of the blood vessels, that means the restriction that whatever is inside a blood vessel should not go out, that is reduced. And therefore, these permeable capillaries, which you see in step 2, they allow an influx of fluid and cells. So cells and fluids should be able to move across the blood vessel and the tissue in order to tackle this inflammatory response. And third step is that phagocytes, they would migrate to the site of inflammation because of chemotaxis that occurs. So upon tissue damage, you see that there is a release of chemotactic and vasoactive factors, like I just said. So because of the presence of chemotactic factors, the cells start migrating towards that region where increased concentration of chemoattractants is present. And then finally, the phagocytes and the antibacteria are able to, antibacterial exudate are able to destroy whatever pathogen had come in. So that is the way in which it occurs. So as an inflammatory response develops, cytokines and other molecular markers that are released, they act on the endothelium of blood vessels. They're able to release these cytokines and vasoactive factors, like I said, and they act on the blood vessel endothelium, which is these cells which are lining the capillary wall. The affected epithelium then becomes inflamed. There is increased expression of certain molecules, certain receptors. You're already familiar with it, which are known as cell adhesion molecules or CAMs. Which can either belong to integrins, selectins, mucin-like proteins, or IGSF, which is immunoglobulin superfamily, which are then able to bind to ICAMs on the endothelium. So there is CAM-ICAM interaction. I'll come to it in the next slide also. But with respect to neutrophils, there are three steps. There are two steps majorly which occur, which is a margination. Margination means that from the circulation, you see these yellow colored cells. These are a schematic representation of neutrophils. From these cells, they are able to adhere to the wall of the blood vessel, which is the process known as margination. And then they're able to extravasate. Extravasate means they're able to, the word extravasate, let's just split it down. Then it means that from the vessel, from the blood vessel, it is moving towards the extravasal side, which is outside of the vessel side. So that is extravasation. The reverse, when something is entering into the circulatory system, into the vasculature, would be known as intravasation. So this is the way in which these two processes work. So the neutrophils will first bind to this inflamed epithelium in the process known as margination, and then they would be able to move out of this blood vessel via the process known as transendothelial migration. So transendothelial simply means that it is across the endothelium, which is what you see here, that the blood vessel entire endothelium is present. And across it, these cells are able to move out, or they are able to extravasate. So like I was mentioning, there are many receptors which are extremely crucial to facilitate this process. So you see interactions between different types of molecules. We'll come to them one by one. Keep in mind here that some abbreviations have been mentioned. HEV is high endothelial venules, which are the site of lymphocyte extravasation. LFA1 is lymphocyte function associated antigen 1. It's simply a protein on the cell surface of lymphocytes. So for this migration or this movement of neutrophils out of the vasculature and towards the tissue that is undergoing damage, this process, which we understood as transendothelial migration, had certain sub-steps, or otherwise it was also called as extravasation. It had some sub-steps. So first step you see is rolling. Second step you see is activation. Third step is arrest or adhesion. People are going on hold for some reason. Anyway, so yeah, so for extravasation, neutrophils must be able to recognize the endothelium that is inflamed. How will that happen? They will release chemokines. The chemokines will attract neutrophils towards the site of inflammation. Secondly, they must adhere very strongly by CAM and ICAM interactions so that they are not swept away by the flowing blood. So like we said, blood flow was increased and vascular permeability was increased. But if there is excessive blood flow, then that would take the cells away, which have already bound to the endothelial lining. So how is that interaction facilitated? By CAM-ICAM interactions, which come in much later. Apart from the CAM-ICAM interactions, we see other as well. So from a naive T cell, you see that there is the binding of L-selectin present on the T cell to CD34, which is present on the endothelial venules. And you see chemokine present here along with ICAM, which is present right next to it. So the neutrophil will start to roll, physically roll, which means that first it binds to L-selectin or L-selectin CD34 interaction is the first one which occurs. Then the neutrophil moves forward a little bit, and upon moving forward, its chemokine is able to bind to the chemokine receptor, which you see is present here. And upon rolling, it comes close. And because of this recognition, because of chemokine and chemokine receptor recognition or interaction, you see in pink that a signal has been triggered, which causes the rearrangement of LFA1, which is lymphocyte function associated antigen 1, that binds to ICAM. So you see it is a series of effects that are occurring. After L-selectin CD34 comes chemokine receptor interaction, which triggers uncrossing or opening up of LFA1, which can then bind to the ICAM1. So CAM-ICAM interaction is occurring, which is then triggering transendothelial migration. And this it is doing after the process of margination, which we had already discussed. So margination is followed by transendothelial migration. So yeah. Yeah. Okay. So this is the way in which inflammatory response is figured out, is first identified and then regulated and then understood. So what you see here are the effectors of innate immunity. So when we talk about the effectors, what we mean to basically look at are that first, after recognition has already occurred and host immune cells have been triggered, there needs to be a response to that information that has been gathered. So the effectors can again be of different types. We see that there are some common principles of innate immune response effector mechanisms. So they rely on certain common mechanisms which have very like superficially been shown here. The reason for keeping it a little more little simplified is because the details of it are already present in the lectures. So what you see are different subtypes of these effectors. The first of which, what you see here in red, are for cationic antimicrobial peptides. Cameras. Okay, so Yeah. So, cationic antimicrobial peptides, which are present here, they are able to, what you see here is an indication of plus charge, which means that it is positively charged. So, what is the reason for cationic peptides being released? It is because a typical eukaryotic membrane is usually arranged in such a manner that the outer leaflet does not necessarily carry a charge, and any negatively charged lipid that might be present is usually put towards the inner leaflet. On the other hand, the charge which is present on microbes or in let's put it as typical prokaryotic membranes, they are very, very rich in charged lipids or negatively charged lipids. So when you have cationic antimicrobial peptides, they're able to bind to those negatively charged lipids, and then they are able to trigger cell death. So that is one particular molecular mechanism of innate immunity. The second one is that you go for antivirulence. When we say antivirulence, it is because of some pathogen which has released some toxins, some proteases, or it can be some antitoxin effectors, etc., which cause damage to the host. Third are the PRR, or third belongs to the PRR assisted elimination or pathogen recognition receptor associated or assisted elimination, whereby you can see that there are multiple immune effectors which are able to bind to our pathogen of interest. There are certain soluble PRR receptors as well, which can do the same. And thirdly, of course, you have lysosomes. So these are PRR, these are responsible for eliminating the pathogen via recognition receptor or PRR. Fourth is infected cells that are undergoing cell death. And this, like I already discussed, this could be something that if is interesting to you, you can read up on it yourself. Else, I will bring it up maybe sometime in the next class itself. So apoptosis and cell death are triggered as a response to that infected cell. Um, Finally, for E, we see nutritional immunity, whereby you have certain between the effector, between the pathogen and the immune response cells that are present. It can be such that the immune effector is triggering the release of certain host nutrients, which are very iron rich, and therefore that is used to uh, tackle invasion by a pathogen. So these are the different effectors of innate immunity. So with this in mind, we will next move to the organs of the immune system. I just need like a minute's break, and I'll be right back. In the meantime, if you have any questions, yeah, sorry, you can raise your hand and I'll just be back in a minute. Okay, sorry for the interruption. Yeah. So we talked about the innate immune system. We talked about the cells, the mediators, the recognition receptors, the effector molecules, and how exactly all that regulation is being controlled. Now, So now we'll come to the next part. So if you remember your lectures, a lot of detail was provided on the way in which B cells and T cells, which come majorly under the adaptive immune system, but which are also operational with the innate immune system. And there is a crosstalk that occurs between the two. They are very heavily dependent upon organs of the immune system. When we say organs, what do we mean? Mainly, there are two organs which are the primary organs, primary lymphoid organs, the bone marrow and the thymus. But apart from that, you have lymph nodes which are present throughout the body. And these lymph nodes are very, very critical because this is how your entire, any individual for that matter, is protected because of the presence of these lymph nodes, because these are the sites where the antigen and the antibodies, or rather the immune system and the pathogen, are able to interact with each other. Yes, somebody has a question. Please unmute and ask.
Ma'am, your slide is not visible.
It's not?
Yeah, just now, maybe just a minute before. Yes.
Okay, I'll reshare. Just
Yes, now visible, ma'am. Thank you.
it's fine now? Okay.
Yes, ma'am. Yes, ma'am.
Okay. So, yeah, so we talk about the organs of the immune system. So, apart from these two organs, the more crucial responsibility is taken care of by the lymph nodes. We'll get into that also in details. So, if you remember the way in which B cells and T cells are produced, the way in which they mature, this entire process has been extensively discussed in the lectures. Again, I'm not going to repeat the same thing, but yeah, I'll give you a little more detail with a more easily available schematics for your own reference so that it's probably a little bit easier to understand. What you see here is a cross section. This is again taken from Kuvi, and this is a older edition. If you have access to the newer one, please refer to it again. And if you need this edition in particular, let me know. I'll upload it in that Google Drive folder that you probably already have access to. So here you see a diagrammatic cross-section of a portion of the thymus, which shows lobules that are separated by connective tissue. So these are the tuberculae, which are these are the blood vessels, which are keeping the contact between the circulatory system and the lymph nodes. So basically, there needs to be a connection between blood vessel, because blood is always carrying oxygen. Every tissue needs to be oxygenated, and there also has to be communication with the immune system. So these two always need to be in touch with each other. So you see the organ is um, is well supplied with blood vessels, which is shown here in pink. And within the organ, we see some sort of a gradient which is present based on the types of cells and the way in which it is organized. Let's look at that. First of all, for any organ, it's a basic anatomy definition that for any organ, like if you take a cross section of it, the outer side of it is always called as the cortex. The inner side is called as the medulla. This is true for any organ, whether it is adrenal gland. There you talk about the adrenal cortex, adrenal medulla. For the kidney, here it's the thymus. For spleen, etc. It's pretty much an anatomical classification. So even for trees, for that matter, you talk about the cortex of a tree, which is the outer layer of the bark. So towards the cortical side, you see a different arrangement of cells, and towards the medulla, you see a different arrangement. Let's refer to the legend for some detail about it. So what you see is that the densely populated outer cortex is thought to contain many immature thymocytes. When we say thymocyte, what does it mean? Very directly means cells of the thymus, right? They are represented here in blue, these little blue cells. These are thymocytes. They undergo very rapid proliferation coupled with an enormous rate of cell death. Now, this question might come to your mind, that why is there very rapid proliferation and why is there such rapid cell death? That is because it's necessary to filter out those cells which would be beneficial for a particular individual to survive and which are important for the immune system to be functional. Now, if you have some background about the energy that, or the ATP, which is the energy currency of a cell, the amount of energy that goes into cell division and also goes into cell death, you would think that this is a very energy intensive process, that a cell needs to spend a lot of resources into dividing and increasing in number and also dying as well. So why is there such a haphazard arrangement which is occurring in these organs? That is because the way in which a living organism functions, there's a difference in the way in which a cell functions and a living organism functions. For a cell, it's just important for that cell to survive. It will communicate with itself and its surroundings also, but to a limited extent. When we talk about a living organism, its priority would be always survival of the entire entity as a whole. Why I'm stressing on this is because this will again bring us back to the definitions that I just mentioned about the different processes of cell death, which was necrosis and autophagy and programmed cell death. So if you read a little bit more about these processes, you will understand that for a living organism, it always prioritizes the greater good. That if for survival of the organism, some cells need to die, it will choose that. So in this case, the priority becomes survival of the individual by having a very comprehensive, very efficient immune system. So the whatever amount of energy might go into generating a B cell or T cell repertoire, which is capable of tackling pathogens of all different type, the body is ready to invest that amount of energy. And therefore, for children in particular, when there is a gradual development of an immune system, the nutrients that go into children are considered to be extremely important. And particularly with respect to the thymus, because at the time of birth, there was a graph also. I don't think I put it here, but you can refer to Kobe. I think it's present in the same page as this thymus cross-section image, where it shows that at the time of birth, an individual's thymus is of the largest size, is or weighs the heaviest because it has that much, that huge a repertoire of T cells or thymocytes, and then over time it becomes smaller and smaller, and our immunity becomes more and more compromised, and people beyond the age of 30 start having deteriorated immune system and are more prone to diseases. So what I'm trying to say is that at the time of birth and during the younger years, a lot of energy is spent into developing a very strong immune system, because a that person is young, that person has not encountered pathogen and all these different sorts of microbes which are present in the environment. So for them to be able to be defended well, energy is spent into developing thymocytes. So there is a huge amount of energy that is invested into dividing cells and creating more and more thymocytes, or generating or encouraging more and more cell division. But at the same time, what you see here represented as these dotted lines are dead cells, which have been removed because either they were specific to the self cells, or they were not healthy, or for a number of other reasons. So the idea of this me giving all this extra information was for you to understand that any biological organism always chooses survival of the entity as a whole and not necessarily survival of an individual cell, which is where when we talk about cancer, for cancer cells, all they care about is their own division. So there, the idea that if one particular cells die, if one cell dies so that another cell can live, it no longer becomes a priority. There, you just want to form a tumor. Those cells are abnormal and they cause disease. That's why it's a very difficult disease to tackle. Now, coming back to the immune system, what we see is on the outer side, it's very well enriched in thymocytes, which are immature and which undergo rapid proliferation and cell death. Also, what is present in the outer cortex are cells which are shown in gray, which are the thymic nerve cells. What are they? They are specialized epithelial cells with long membranous extensions surrounding as many as 50 thymocytes. What does it do? As the name indicates, nerve cells are those which are responsible for nourishment of other thymocytes. When we say nourishment, it means because they are immature thymocytes, for their they provide certain factors, certain cytokines, etc., which help in proper growth and proper division of thymocytes. So these you see are much longer with sort of extensions, and they surround a large number of thymocytes and help in their better growth. On the other hand, when we talk about the medulla, it is relatively much more sparsely populated and is thought to contain thymocytes that are more mature. So the outer side has immature thymocytes. On the other hand, the inner side it's a lot more, it's not as densely populated with cells, and it contains more mature thymocytes for that matter. So during their stay within the thymus, the thymocytes interact with various stromal cells, including cortical epithelial cells, which are shown here in light red, medullary epithelial cells. So these are different types of cells. I'm not going into each of them individually, interdigitating dendritic cells and macrophages, etc. The question is, why is that important? That is because these cells produce some thymic hormones, and more importantly, they express high levels of class 1 and class 2 MHC molecules. If you remember the lectures for this week, that it was explained that during the process of maturation, for the definition or for the um, distinction of a particular thymocyte into CD4 plus or CD8 plus, interaction with MHCs is important. Now, who supplies those MHCs? These cells, which are part of the population that pre-exists within the thymus. So all these different types of cells that you see, these macrophages shown in yellow, the medullary epithelial cells, the interdigitating dendritic cells, or the cortical epithelial cells for that matter, all of them have certain MHCs on their surface, and based on the type of MHCs that they carry, which is something we will also come to when we discuss MHC in more detail, because each of these types of cells has different receptors on their surface. Again, more advanced concepts, we'll come to it later. Based on the type of interaction that these thymocytes undergo, they will differentiate into CD4 plus or CD8 plus cells. Apart from that, Hassel's corpuscles, which you see here shown sort of like a network like structure, much like the endoplasmic reticulum or Golgi, it looks a little bit like that. So these Hassel's corpuscles are found in the medulla, and they contain concentric layers of degenerating epithelial cells, and they don't, at least from what I recall, they don't necessarily have to have a role to play, but instead they are epithelial cells which are slowly dying, or they're forming these distinctive structures, but they're quite visible in the thymus, which is why you can see it in the cross section. So this is how the thymus looks. Now, this will we come to the next organ, which is what I said is the bigger contributor towards sustaining the immune system at large, which is the lymph node. Again, you are familiar with it already. You know how it works. You know how it is very, very crucial towards the working, or towards the antigen antibody interaction, or towards the sensitization of the immune system to the pathogen that has come in. But let us go through the structure of it first, so that it's a little clearer. So what you see in the inset is more important in this first figure. So these are the three layers of the lymph node, which have very, very distinct microenvironments, and which is what is crucial towards the way in which development, or the way in which its job as part of the immune organ can function. So, of course, as we already said, the outer part is the cortex, and then a part is the medulla. In this case, there is another region which is somewhere between the two, which is called as the paracortex. So that is a broad classification of the way in which the lymph node can be looked at. So this is a simpler representation of a lymph node, and then we come into the more detailed structure of the lymph node. So here I'll read out the legend so that we can refer to it simultaneously with the figure. In the bigger picture, what you see here is that the left side, it depicts the arrangement of reticulum and lymphocytes within various regions of the lymph node. Macrophages and dendritic cells. What are those? You already remembered that they are antigen-presenting cells. What do? They trap antigens, which is why they're able to present it later on. They are present in the cortex and the paracortex. T cells, H is in subscript, or T helper cells. They are concentrated mainly in the paracortex. Then come B cells, which are located primarily in the cortex, particularly within the follicles and germinal centers. You already know what are germinal centers, where rapidly proliferating regions, which are very rich in these B cells. So this is how the localization is carried out. So let us just look into it for reference once again. The cortical region, or better here, the yellow colored cortical region, is very rich in macrophages, dendritic cells, and B cells. All three of them are antigen presenting cells. Keep it in mind. Then come the T helper cells, which are present somewhere towards the middle, the orange colored part, which is the paracortex. And B cells in particularly within the cortex, they are present or they're concentrated within the germinal centers and follicles. The medulla, on the other hand, is populated largely by antibody-producing plasma cells. If you remember, as we had discussed in the previous class and also in the lectures, you know that plasma cells are just the activated B cells, which are now producing antibodies or producing secretory antibodies, which will be released into blood circulation, lymphocytes that are circulating in the lymph are carried into the node by afferent lymphatic vesicles. If you have studied the circulatory system before, when I say circulatory system, I mean the way in which the heart and the aorta and arteries, ventricles, etc. operate. It is kind of similar to that, that you have some systems, some blood vessels which are bringing deoxygenated blood into the lungs, purifying it, and then sending it to the heart, and then that oxygenated blood is being pumped into the system. So that's how the double circulatory system used to work. Similar to that, for the immune system, this is what a lymph node is also kind of. It's not pumping out anything fresh in this case, but yes, there are certain channels for input and certain channels for output as expected. So the afferent vessels, which is what is mentioned here, the afferent lymphatic vessels particularly, are responsible for bringing the lymphocytes that are circulating in the lymph nodes. Circulating in the lymph. Lymph is the fluid which is circulating throughout the lymphatic system. And from there, afferent lymphatic vessels will be responsible for its entry. So that is what you see. I'm not sure if it's visible. Yeah, so this is what is bringing them in. And then what you see, they either enter the reticular matrix of the node or they pass through it and they leave by the efferent lymphatic vesicle. So either they will enter here, and if they don't encounter their specific antigen or their cognate antigen, cognate is the term which is used in particularly for immunology. Cognate means it's specific. So a receptor has a cognate antigen. And antigen antibody interactions are occurring because they are cognate or they are specific for each other. It's just the semantics, the correct terminology that is used. So either it can enter through the afferent lymphatic vesicles. If it doesn't encounter anything, it will exit through the efferent lymphatic vessel, as you obviously can see. Also keep in mind that you see there are multiple afferent lymphatic vessels, but only one exit vessel, because again, that is the way in which the structure prioritizes the fact that if there is an infection and you need to bring lymphocytes, you should have multiple routes of entry. Upon interaction, if nothing is happening, then exit route can be minimized to one. So the right side, which is shown here, it represents or it depicts the lymphatic artery and vein and the postcapillary venule. So this is showing you the cross section where the types of cells that are present can be seen. So you see these regions within the cortex, which are those germinal centers that we talked about, which are very, very rich in B cells, which are very active sites of proliferation for B cells as well. So these are the germinal centers with B lymphocytes at the central region. And you also see these primary lymphoid follicles which are present. And towards the paracortical region, these blue cells are representative of the H cells, like I already said. And this side is showing you the exit entry route. So lymphatic artery and lymphatic veins are present, postcapillary venules are present. Lymphocytes in circulation can pass into the node from the postcapillary venules by a process called extravasation. We already talked about extravasation previously. So that's just giving you a bird's eye view of the process that we have already discussed in detail, that if there is infection and there is a need to move out from the blood vessel, then this is how they'll do it. Until it is activated by an antigen, a lymphoid follicle, which is called as a primary follicle, depicted here. It comprises of a network of follicular dendritic cells and small resting B cells. So what you see in these two cross sections is a region where it's not very actively proliferating, and it mainly comprises of dendritic cells and resting B cells. When there is an antigenic challenge means that there is an antigen which has come in and which needs to be tackled at that point of time because of an infection that has set up within an individual system, then a primary follicle becomes a larger secondary follicle, and there you see the concentrically packed B lymphocytes surrounding the germinal center. And at the center, you have very rapidly proliferating B lymphocytes and an area that contains non-dividing B cells, helper T cells interspersed with macrophages and follicular dendritic cells. Again, this is a lot of information, but it's just what the primary and the secondary lymphoid follicles are comprised of. So you should have the information on hand, which is why I've kept all of it. But for conceptually, you need to understand where these are located. Why these are located at the cortex? It's because after it has entered through the lymphatic vessels, it should be able to encounter B cells first. With the help of T helper cells, it will stimulate the T cell mediated immunity, and then both the branches together can tackle whatever pathogen has come in. So this is the overall organization of a B cell. Just put it on charge. Yeah. Sorry for the interruption again. But yeah, so we were talking about this lymph node and we wrapped it up. Now, after all that discussion about B cells, what we now look at is the way in which B cells develop. So if we look at, if we take an overview of the way in which B cells develop, so if we look at the overall process by which B cells develop, then you see there are two phases, broadly two phases, an antigen independent phase and an antigen dependent phase. During the antigen independent phase, what simply happens is that from all the B cells that we saw within the germinal centers, from the resting or the immature state, they will undergo a process known as gene rearrangement. Again, we are not getting into the details of that right now, but IgG gene segment rearrangement occurs, which gives or which annotates one particular specificity to all the B cell receptors which are present on the surface. If you remember correctly, then I'd mentioned that approximately 10 to the power 5 BCRs and TCRs are expressed on these cells respectively. So after that gene segment rearrangement has occurred, then there is selection and there is maturation into B cell of a particular lineage. Now, from there, you see that approximately 5 into 10 to the power 6, which is around 5 million cells per day, are coming into the peripheral lymphoid organs. What happens there is that you see that this is a naive B cell. So again, these terminologies are very important. Mature B cell means something which has already undergone gene segment rearrangement and it is specific for a particular antigen. So its surface has receptors which will bind to one particular antigen. That is a mature cell. But it is naive. Naive means that it has not yet met its antigen. So it has not undergone activation. It has not faced its cognate antigen. So that is a naive B cell. If for an extended period of time it is in circulation and there is no antigen, this is minus antigen means it does not encounter its antigen, then that cell will eventually die. It means that that particular B cell, while was initially made to be useful, has not encountered its cognate antigen and is no longer required and undergoes cell death. This happens in 90% cases. Again, this highlights how important and how well prioritized the immune system is and the amount of energy that is invested into making these cells and going through the process of gene segment rearrangement, giving them a specificity and then simply allowing them to die. That is because it's more beneficial for those cells to die and for those resources to be reutilized in a different direction rather than making more and more of those cells which are not specific. On the other hand, the remaining 10%, which is a very, very small population, does encounter its antigen. When it does so, with the help of T helper cells, which again, if we just to revise, T helper cells were present in the paracortical region of the lymph nodes. Those T helper cells allow activation of the naive B cell towards the into the activated B cell. So from the naive B cell, it binds to its antigen and it becomes an activated B cell. When activated, it can undergo or it will undergo two types of things. There would be class switching. Class switching is a process, I think it was discussed in the lectures as well. Class switching is a process where subtype of antibodies can switch based on the type of response which is required, which is IgA, M, E, T, and G. So among them, there can be changes. And on the other hand, they undergo a different process known as affinity maturation, which I'll briefly discuss in another schematic. But yes, more details about it is something you'll learn after gene segment rearrangement. But after undergoing these two processes, from the activated B cell, they can either form, not either, they will form plasma cells and memory B cells. So plasma cells will be those which you see they have a well-defined ER and, all of the organelles in general, and they will generate secretory antibodies or secreted antibodies, which can go into circulation. On the other hand, some cells will be retained and they will serve as memory B cells, which will be useful in case that same person encounters the same antigen sometime in the future, so as to mediate the immune system memory response. So that's how this works. Let's go over the legend once, just to make sure we've not missed out on anything. During the antigen-independent maturation phase, immunocompetent B cells expressing. Immunocompetent means that they are capable of eliciting an immune response. Please keep these terminologies very clear in your head. Immunocompetent is something, in fact, it does not even necessarily need to be a cell. It can even, you can even refer to a person as immunocompetent or an immunocompromised. When somebody has the capacity to respond or has the capacity to elicit an immune response, then they are immunocompetent. When they do not have that ability, they are immunocompromised. So when people are given drugs for transplantation, before transplantation, immunosuppressive drugs, then such people become immunocompromised, or they might have some disease which causes that condition as well. Particularly, I work in the field of cancer biology, so we work extensively with immunocompromised mice. So they basically do not have they do not have an immune system. Different branches of their immune systems are compromised. So you can, if it is something that is interesting to you, you can read up on it, how immunocompromised mice work. Like there are subclasses where some of them don't have B and T cells, some of them don't have NK cells, some of them have B cells. So they have different systems. So they're actually quite, it's interesting. You see not skid gamma mice and so on and so forth. Again, without deviating too much, because we still have some information to cover. Only 10% of the potential B cells that, like we just saw, are able to reach maturity and they're able to exit the bone marrow. Naive B cells in the periphery will die within a few days if they don't encounter their soluble protein antigen, which is this minus antigen condition, and activated Th cells. Once activated, in case they have met their antigen, and of course the supporting T helper cell has also met the same antigen, then in that case, what happens is that B cells will undergo proliferation. This process is also often called as clonal selection, which is for selection of a particular clone of B cells. Why are they getting selected? Because they are specific towards an antigen which is present on some sort of foreign pathogen which has invaded into the body. Those bearing high affinity, MIG stands for membrane immunoglobulin, or surface receptor, let's call it that for now. Those which have very high affinity, which is a process of affinity maturation, those which have high affinity antibodies will then undergo the process of differentiation into plasma cells and memory B cells. And these can instead also express different isotypes. Isotypes means the different subclasses of antibodies that I talked about. There are 5 subclasses, IgG, IgA, IgM, IgE, and IgD. You will read up on this. There's lots of information to cover. Don't worry. For now, just understand the concept that during the process of B cell maturation and B cell development, all these sub steps occur. That if antigen is encountered, then such a B cell will be selected for, and such a B cell will undergo class switching and affinity maturation to form activated B cells and memory B cells. So this is the process that is representative of what happens in mouse, but the same principles they occur in humans as well. So why mouse? Because mice are the most convenient and most widely used models for studying any human process, even today. So that is the process of B cell development. And within the germinal centers, this again is, I'm not getting into extensive detail about this process. This is exactly what Professor Ganguly has explained in his lecture as well. So I'll keep it a little brief, but yeah, we'll still brush up on the basic principles that occurred. It's just a better schematic, I think, for you to understand. It's more organized in some way. So this schematic, also again taken from Kubi, is a representation of the events that occur within the germinal centers. So again, you remember the germinal centers are regions where there's very rapid B cell proliferation, right? And it is present within the cortical region of the lymph nodes, of the peripheral lymph nodes. So there, what you see first is that, let's go by the legend only because it's more organized. So antigen stimulated B cells migrate into the germinal centers where they reduce the expression of surface Ig, surface immunoglobulin, or otherwise also known as membrane immunoglobulin, and they undergo rapid cell division and mutations of the rearranged immunoglobulin regions within the dark zone. Let's not get into the rearrangement part right now, but let's just understand what is happening with respect to the principle for now. Subsequently, cell division stops, and the B cells will migrate into the light zone and increase the expression of surface immunoglobulin. At that stage, what are they called? They're called as centrocytes. At this stage, when they are centrocytes and they are present within the light zone centrocytes interact with the follicular dendritic cells. Here you see this sort of purplish or lavender, this octopus-like structure that you see, that is the dendritic cell. The name comes from the fact that it has these extensions which are known as dendrites. If you've studied the structure of a neuron, you would be familiar with it, that there are dendrites in neurons as well. So this dendritic cell is what sensitizes them. And within this light zone, upon interaction between the centrocytes and the follicular dendritic cells and the T helper cells, they are able to survive. The follicular dendritic cells bind to antigen antibody complexes along their long extensions, and the centrocytes compete with each other to bind to the antigens. Those B cells which bear very high affinity membrane immunoglobulins, like we talked about the process of affinity maturation, they are selected for. And those are which are shown in blue. I think I can't see the color right here. But anyway, conceptually, let's understand it for now, is that those cells that have high affinity will be able to compete successfully. Those that fail this antigen-mediated selection will be, here it is demonstrated in black, they would die by the process of apoptosis. Now, B cells that have passed this process of antigen selection is performed by the follicular dendritic cell, which is an antigen-presenting cell, which is displaying antigenic peptides on its surface via MHCs. So B cells are able to interact with those antigens. If they are able to bind, or if they're able to interact with very high affinity, then they are selected for. If they have low affinity, they would undergo apoptosis. So those that pass the antigenic selection, they receive, and those that receive a second survival signal or a supportive signal from T helper cells will then differentiate into memory B cells or antibody secreting plasma cells. The encounter with T helper cells, like we already discussed, might also induce class switching. And a major outcome from the germinal center is to generate higher affinity B cells, which here has been represented as Ka2, from B cells of lower affinity. So the initial exposure generated B cells that had low affinity, which is at the bottom is represented as Ka1. Ka is just it's a constant for affinity, that if you could measure affinity of you can actually measure affinity of antigen and antibody with each other. So these were low affinity B cells. upon encountering a pathogen, they have come back. And within the dark zone, first there is a somatic mutation, very rapid mutation, which is occurring within the centroblasts or the activated B cells, which is generating a huge repertoire of B cells that all have different affinities to the same antigen. If it is confusing, please let me know. I'll try to simplify it a little bit more. So think of it as I don't know how to explain it. Think of it as a bottle. This bottle, for example, if this is your antigen, what it will do is initially the B cells that have come in, they are specific for this particular antigen. Now, after entering into the dark zone, what will happen? B cells which are specific for this antigen are undergoing rapid mutations. These are mutations that are beneficial and that are necessary so that from that same B cell, which had one particular specificity, it is generating B cells that have very different types of specificities. Some of them might bind here, etc., with different affinities. Now, the next priority comes the affinity. Affinity means how strong or what is the strength of the interaction of that antibody with that antigen. So somebody might be able to bind more strongly, somebody might have less strength, or somebody might completely lose their specificity, so on and so forth. There are different combinations which can be generated because of these mutations that are occurring. So from this somatic mutation which are occurring in the dark zone, what happens next is that from that entire repertoire, selection needs to be carried out. So you have, say, from initial one type of antibody, you have 10,000 antibodies, or 10,000 different types of antibodies. From these 10,000 types of antibodies, only those which have high affinity needs to be selected. How will that happen? The follicular dendritic cell, which is already an antigen presenting cell, will display the same antigen, the bottle that I showed you, and then it will be the different types of B cells that have been generated will bind to it. Those that have high affinity will be given a green signal. Those that do not have high affinity will die. From this population, say from 10,000 cells, we have narrowed down to 1,000 cells. From these 1,000 cells, even further filtering is carried out. How? Then comes the role of T helper cells, which we already discussed, are present in the paracortical region of the lymph node. Sorry. So within the paracortical region. So T helper cells will provide additional stimulus, and they will select specifically for those cells that have been identified to be specific for the antigen. Once this added stimulus is taken into consideration, then class switching can occur, like we already saw in the previous diagram. This is just the same process. This process has been explained. This one step that you see so briefly shown here, that entire process occurs with all this detail. It's just an expansion of what is happening there. Don't get confused. It's just more detail because you're learning concepts in more depth. So upon this selection, there can be differentiation into memory cells. Memory cells will move out, will go into circulation and remain as an immune response, backup immune response. And on the other hand, plasma blasts, or otherwise they are known as plasma cells, will produce antibodies that will be secreted. And now these antibodies are of higher affinity, which was the entire purpose of going through this huge process. So I hope it is clear what is the objective of B cell development, what is the objective of going through this entire process. So now you have antibodies with very high affinity, which will be able to bind quickly and strongly with the antigen and will be able to clear it off as soon as possible. I think we only have a few more slides left. Yeah, just this is the last slide before questions. I have not gone into the details of T cell development. It's already explained. I've just gone through the selection process that occurs for CD8 and CD4 plus. So if you remember, once again, the cross section of the thymus, we saw how the arrangement was performed. So in immature thymocytes, let me just go back to the figure for easy recollection. Yeah, so immature thymocytes were on the outside towards the cortical region along with nerve cells, whereas the cortex, whereas the medulla by itself was a lot more sparsely populated, but it was very rich in the antigen presenting cells, or APCs. So with this in mind, what happens is that T cell precursors, they arrive at the thymus from the bone marrow via bloodstream, and then they undergo development into mature T cells. Then they are exported to the periphery, where they can undergo antigen-induced activation and differentiation into effector and memory T cells, much like the process that you see for B cells as well. But this process is, while it seems very simplistic, it occurs in different regions and it occurs with a lot of complexity as well. Each stage of these events is characterized by some state-specific intracellular events and display of some very, very, very specific cell surface markers. Let's come to it. This is the last topic. We'll take it clearly. So first you're looking at the bone marrow. Let us start with the region. Within the bone marrow, you have hematopoietic stem cell, which is the original progenitor cell that gives rise to all these blood cells that we are talking about, right? So it's a pluripotent stem cell also, like we had discussed in the previous class. From there, a certain subclass will form the common lymphoid precursor. If you remember, we had seen an entire expansion of the way in which lymphoid progenitor, myeloid progenitor, lymphocyte, megakaryocyte, etc., all of those differentiation, different subtypes we had studied in the last discussion session. So within the marrow, you have the common lymphoid precursor which has been generated. This precursor will migrate from the bone marrow via blood into the thymus. So this is all the part that you see for the marrow. From there, it has to move into the thymus because it's a T cell. Its maturation will be completed within the thymus itself. What you see here on the right is a representation of the surface markers. You don't need to remember this much detail right now, but this is just for you to understand how these cells keep on evolving. So when it is a stem cell, it has expression of certain markers like CD44, CD25, c-KIT, etc. But when it is maturing over time, you see that its expression of some of these markers is lost and new markers come up. So each sub-step or each of these steps can be distinguished based on the type of cell surface marker which is present in each case. Coming back to the process which it is undergoing, from the common lymphoid precursor, once it has moved into the thymus, it is considered as a T cell precursor because now it is destined or it is pre-programmed to differentiate into a T cell. It cannot develop into a B cell anymore. Within the thymus, then the next step is the formation of some or the subclassification of these T cells into CD4 plus or CD8 plus. So what you see on the right, you can avoid it for now because this is the entire process of gene segment rearrangement and it's complicated. You don't need to go into it. It is BDJ recombination. Let's avoid it for now. But it's basically just to keep it in mind. It is the rearrangement of the TCR, the T cell receptor. If you remember from our previous class that B cells and T cells have their own receptors. B cell receptors are antibodies. T cell receptors are TCRs. I'll just go back to that slide. Yeah, so these blue entities, these are antibodies, which are B cell receptors. These are TCRs. CD4 and CD8 are co-receptors. The process that we are discussing right now is the maturation of T cells to either become CD4 plus T helper cells or CD8 plus cytotoxic cells. The TCR rearrangement is something completely different. The TCR rearrangement is for a particular antigen, and that rearrangement is more complicated. We'll come to it much, much later. I just wanted to clarify this. Right now, we are just trying to understand CD4, CD8 development. So coming back to this slide. So from the T cell precursor in the pro T cell stage, it is initially double negative, which means it is CD4 minus, CD8 minus. It does not express either of these two markers. Then from the pro T cell, the differentiation between the pro and pre T cell is that it has undergone this TCR rearrangement, which takes place first. So again, we are not going into details of it now, but these two processes occur simultaneously, which is why the schematic is representing it. So it undergoes this recombination and then it is a pre-T cell and it is still in the double negative state. From the double negative state, then it progresses towards the double positive state, which is again a pro-T cell. But the difference between these two is that it is double negative, it is double positive. When we say double positive, it means it has both CD4 and CD8 markers present on its surface in this particular stage. Keep in mind that here you see that surface markers are changing. Before this, you do not see CD4, CD8 expression. From the pro-T cell stage, you are able to see double positive expression. Simultaneously, you are also able to see some other TCR chains you can avoid it for now because again, complicated concept. But CD4, CD8 expression starts at this stage. From here, there is differentiation into either of the two subclasses. It does not happen in it is ideally at least not supposed to ever happen in this manner that any double positive cell will go into circulation within a system, because that can wreak havoc. Both of these cells have very distinct functions. They operate in different ways and they mediate different branches of immunity. So from the double positive cell, there absolutely perfectly has to be selection into either of the two subtypes. Either they'll be CD8 plus or they'll be CD4 plus. That is a separate process. We can get into a little bit detail about it maybe in the next class if we can spare time. But after differentiation is occurring into either of these two subtypes, they will then move out into circulation. When they move out, this is a TC cell, which will go into peripheral tissues and it will directly be able to cause cytotoxicity. It will be able to kill cells directly. On the other hand, the CD4 plus T cells, which are helper T cells, they will connect the B cell mediated immunity, which is the antibody regulated humoral immunity with the T cell mediated immunity. It will be able to link the two and will be able to convey that signal. And why that is important is something we just discussed in the previous slide, that once B cell selection, first round of B cell selection has been carried out, it is the T helper cells which are coming in and providing an additional green signal that yes, this is a better antibody and you need to produce more and more of it. So it is helping and mediating the immune response for both B cell and T cell mediated immunity. This is the information I wanted to give for today. There is always more to learn, but that is the idea we wanted to look at. Now just we'll go through a few questions from previous years so that you can get an overall overview of the concepts. So the first question is that T cell mediates humoral immunity, non-specific defense, cell-mediated immunity, or none of these. The correct answer for it would, of course, be that it mediates cell-mediated immunity. It also regulates humoral immunity, but does not do so directly. So that is the job of T cells. The first acting cells of the immune system are B cells, T cells, neutrophils, or NK cells. If you remember that the first line of defense for any wound injury, etc., was controlled by neutrophils. They are the first ones to rush at the site of injury. So the correct answer for this would be neutrophils. NK cells also have a major role to play in the innate immune system. And of course, B and T cells are majorly part of the adaptive branch of immunity. So they come in much later. Pathogen-associated membrane patterns, or PAMPs, are recognized by PRRs, PRPs, PRAs, or RRRs. Of course, among them, these are pattern recognition receptors, or PRRs, would be the correct answer. Then question 4 is that in a macrophage, phagosome and lysosome is fused to form a phagolysosome. What happens after that? This is something we had discussed in the previous week, not today, but anyway, it's an overall part of the course itself. So once phagolysosome formation has already occurred, what happens next? It is degraded and processed peptides are displayed on the surface. It remains intact inside the cell. It is released as it is from the cell, or all of the above. So keep in mind the question is talking about a macrophage in particular, which, if you remember, are antigen presenting cells. So they will chop up the peptides, and those processed peptides will be displayed on the surface via MHC molecules. So this is the correct option A. As the dendritic cells and macrophages migrate to the lymph nodes, they would now be able to interact with which of the following? Mature B and T cells, naive and B and T cells, APCs, or NK cells. So dendritic cells and macrophages themselves are the antigen presenting cells. Obviously, they don't interact with APCs or even with NK cells. And when they are migrating to the lymph nodes, they would be interacting with mature B and T cells. Naive B and T cells are present within the bone marrow and the thymus. Mature cells are released into circulation and they are present in the lymph nodes. And this is very crucial because if immature cells are there, they would not be able to interact with the antigenic peptides, which are displayed by dendritic cells and macrophages. So the correct option is mature B and T cells. Then in question 6, the cell adhesion molecules, or CAMs, include, okay, I did not show this today, but it was part of the lectures. If you remember, mucin, selectins, integrins, or all of the above. The correct answer is all of the above. All of them are cell adhesion molecules that are of different subtypes. We talked about selectin today, but integrins are also cell adhesion molecules and so on and so forth. So all of the above is the correct option. In question 7, the correct order by which neutrophils are recruited at the target site is which of the following? This is a process we discussed in detail today. So first step was, of course, rolling, if you remember. Then there is adhesion, activation, transendothelial migration. This would be the correct option. That is rolling, adhesion, activation, and transendothelial migration. Then in question 8, migration of neutrophils by crossing the HEV blood vessels is known as extravasation, endocytosis, exocytosis, or phagocytosis. Again, a process we have discussed in detail today. It is the process of extravasation, very tightly regulated process where neutrophils will pass through the between the endothelial cells that line blood vessels. And in fact, there is one specific name for that process as well. It is known as pinocytosis, where they move through, where they sort of squeeze out through the space that exists between those cells. So that is what occurs there. And in the last question that we have for today, the double positive T cells expressing both CD4 and CD8 co-receptors are undergoing selection by which of the following? The thymic epithelial cells, the endothelial cells, the lymphoid endothelial cells, or none of the above. So from the CD4 and CD8 co-receptor cells, it is the thymic epithelial cells. Just a little uncertain about it. Yeah, it should be thymic epithelial cells that are undergoing the selection that occurs from the CD4, CD8 double positive cells into single positive CD4 or CD8 co-receptor containing cells. So yeah, that will end the topics that we have for today's class. So if anybody has any questions, please go ahead. I know this was a very information intensive class, but if you have anything to ask, please go ahead. Yeah, somebody has raised their hand. Please go ahead.
Ma'am, just wanted to know that what are these lymphoblasts? Lymphoblast.
Right. So whenever terminologies include the term blast, it means those precursor cells which undergo very rapid division. So from the lymphoblast, you will get lymphocyte. It is a cell that has more controlled division. From the lymphocyte is a more, like you can think of it as a precursor and a differentiated state. So for example, if you have heard of see, the centroblast, for example, when we saw that process of thymic differentiation, so the centroblast was something that was undergoing very rapid division. From there, you had some specific cells, they were a lot more controlled. Even for bone marrow, you have cells that are very rapidly dividing. They are osteoblasts and there are osteocytes. So osteoblasts are those which divide very rapidly. Osteocytes are those that have already divided. So it's just a precursor, the rapidly dividing precursor. Did that clear up your question?
Yeah, yes, ma'am. So I understood osteoblast and osteoclast. So it basically for the multiplication.
Yeah, a cell that is undergoing more rapid division would be in the blast stage. It's a progenitor or a precursor.
Okay, ma'am. Thank you, ma'am.
Any other questions? You can also upload your questions on the discussion forum, or you can ask them, of course, in the class itself. If not, then I think I'll end the meeting for today. The video recording will be uploaded for your reference and you'll have the slides also for access. Please refer to books because no matter how much is taught in lectures or in the discussion sessions, it's still not enough. You need to study it yourself for your own understanding. So yeah, with this, I think I'll conclude the class for today. Thank you so much for joining. If there are any doubts, please come up with them in the next class. I know I gave a lot of information today. please let me know if it was not clear, if more information or more simplified information is required. Refer to the textbooks and refer to the lectures particularly, because they have been explained very well. All the concepts have been explained really well in the lectures. So thank you so much for joining, and have a good day. I'll end the meeting now. Thank you.